In this video, you will learn about protein packaging. Proteins are important macromolecules to the human body. For example, our body requires 20 amino acids to function properly. In addition, proteins play a key role in metabolism, cell signaling, transport, structure, movement, and defense against disease-causing agents. Before protein synthesis can occur, the component parts of the ribosome must be assembled.
Throughout the process of transcription, DNA is transcribed to mRNA inside the nucleus of a cell. RNAs are the product of many different genes. After transcription, the mRNA leaves the nucleus and goes into the cytoplasm. It then goes through the process of translation, where initiation and elongation occur before the protein is packaged.
Then the rough endoplasmic reticulum, also known as the rough ER, compartments that are connected to the nuclear envelope, function to synthesize proteins that will be secreted out of the cell. The rough ER is part of the protein packaging process, not the smooth ER. Let's compare and contrast key details between the rough ER and the smooth ER to avoid confusion. The rough ER has embedded ribosomes on its surface, while the smooth ER does not, hence the name smooth. Smooth ER is primarily involved in lipid synthesis, such as steroid hormones. It also modifies carbohydrates, breaks down toxins in the cells of the body, and stores calcium.
The rough ER secretes proteins that will go to the Golgi apparatus by the pinching off of the vesicular sac from the rough ER around the protein. The Golgi apparatus is made of cisterna flat membrane sacs. There are two sides to the Golgi apparatus, the cis and the trans side. The cis side is used for receiving the protein, while the trans side is the shipping side. The Golgi apparatus is similar to the postal service, where it takes in mail, proteins, and then sorts and ships them to their destined location. The proteins can travel to various locations, including the cytoplasm to stay in the cell, or the cell membrane to exit the cell. Vesicles come from both the smooth and rough ER. Secretory vesicles fuse with the plasma membrane during secretion. These vesicles are made in the rough ER and transported to the plasma membrane to be secreted. Transport vesicles bring phospholipids and proteins to the cytoplasm to remain inside the cell. For example, these vesicles transport proteins and lipids to be embedded in the plasma membrane, forming the fluid mosaic phospholipid bilayer model.
The Golgi apparatus can be comparable to a post office. In this skit, we will demonstrate this concept. In this post office, we have two sections, the receiving side and the shipping side. The receiving side, or the cis side of the Golgi, receives packages to be sorted out according to their destination. Once the packages are sorted out, they are sent to the shipping side, or the trans side. The shipping side is supposed to mail out the packages to its rightful destination. I am the postal office worker who delivers mail to the city. My co-workers label the packages and passes them along to me so that I can personally deliver the packages to where it should go. This could be compared to how the Golgi packages proteins and directs vesicles to either the cytoplasm or the cell membrane. What's interesting about this process is that from the rough ER, COP2 vesicles travel along microtubules to the cis side of the Golgi. As the proteins move through the Golgi, different enzymes are used to affect the functionality of the protein. When the package or protein has been modified for its needed function, the proteins are targeted to their specific transport vesicle. Proper sorting is thought to be directed by the specific amino acid sequences in the proteins themselves. As the proteins begin to collect in their vesicles, unique integral membrane proteins are included called V snares. These proteins working with other integral proteins, such as SNAP25 and T snare, work cooperatively to specifically bind a particular protein to its destination.